this morning to introduce our speaker, who is Mihai Curtasu, who is currently a postdoc researcher at Aarhus University Department of Animal and Veterinary Science in Denmark, and he will present his work this morning. So thank you, and the floor is yours, Mihai.
Thank you very much, Clemence, for the introduction. I will talk today about pig models for human nutrition and also about metabolomics techniques and how they can be used for exploration, or how I've used this technique for exploration in my studies with the pigs. So pigs have a lot in common with humans at the level of organs, genetics, body functions, and the immune system. So using pigs in research studies can predict similar results in humans. And of course, there's many situations or examples, but one out of these many is, for example, the heart of the pig, which is very similar to our own in terms of size and structure. So pigs are very relevant to human cardiovascular research and can even be suitable for the use of heart transplant research. Also, for example, in toxicity study or like drug development studies to approve a new drug for human use. There's regulatory bodies who demand to provide tests, like test data on 2 different species of research animals. So usually the first one is a rodent. They're used a lot in this type of research. But then also larger animals, and most of the time it's either monkeys or dogs. But nowadays the pig is becoming a more viable option. And for example, with some specific drugs, pigs are more tolerant than rodents or dogs, and they can be more representative for the type of reaction that's expected in humans. Because of these similarities, a lot of models have been researched until now. And for example, as I mentioned earlier, the heart similarity has led to the development of cardiovascular models in myocardial infarction, for transplantation, for stent implantation, arteriosclerosis. Of course, in nutrition, looking at the healthy or unhealthy state, so obesity, induced colitis, IBD, or ulcers. A lot of uses for the pigs in biomedical research is on dermatological models. So their skin is very similar to ours, and there has been a lot of research done on wound healing, on surgery, plastic surgery techniques, reconstruction, skin grafts. As well, the kidney of the pig is very similar to ours, so there's been a lot of models on transplantation or on hypertension, this intrarenal surgery. And also there has been some studies done on for the eyes. So the ophthalmological models, because the human eye is the pig eye is very similar to the human eye. Of course, there's also some disadvantages, and these pig models don't come like cheap unless it's a conventional breed type. Those are, let's say, cheaper, and you can have more animals. But if you want to use some very specific type of pigs, and in this case, I'm talking about the miniature pigs, these cost a lot more. They also have, you can find less numbers of it for some specific breeds. And they come with an increased maintenance and also the need for special handling. So some specific breeds, the miniature breeds, and also when you want to have a clinical setup, so you want to do some specific intervention, some sampling procedures, that's when they become a bit more difficult. There's also some differences sometimes in how the physiology works. Like you cannot induce insulin resistance through the diet in miniature pigs, and most of the time that is done with some chemical induction, like a streptozocin, which kind of destroys the pancreas and allows for an insulin resistance. The primary fatty acid synthesis in site in pigs is the adipose tissue, whereas in the humans, that's the liver. when it comes to pig and nutrition, I strongly recommend looking into this review that was published in 2016 that covers very well the aspects of the pig model for human nutritional physiology. But basically, due to these anthropometric, so similar in size, dietary, and also the GIT anatomy and physiology, the pig is a very valuable model. Because what we eat can influence our risk to certain disease, like for example cardiovascular diseases or cancer or microbiome dysfunction. Pigs can provide vital clues to healthy but also unhealthy lifestyles, and it allows us to study conditions related to diet, such as obesity and diabetes. And also, as a large animal that can be fed very similarly to humans, the pigs are well suited for nutritional studies and researching the microbiota. So I'll give here a few examples from studies done at Aarhus University where human and pigs were fed exactly the same bread of different types with different fiber levels. And you can see here the response to white bread versus rye bread, which has very high levels of fibers. In humans and in pigs, it was very similar response. So a white bread that has very easily and very highly absorbable and digestible carbohydrates gives a high peak of glucose in the postprandial response, whereas a bread that's high in fiber, the peak of glucose is much lower. And this has been also seen in pigs as well with the same response, where you have this dotted line, much lower response for the rye bread. Even further along the digestive tract, we see similar responses. So in these pigs, where a Western style diet was compared to a resistant starch diet and to a arabinoxylan rich diet, we can see that the resistant starch and the arabinoxylan rich diet has increased the fermentation in the hindgut, which has led to the production of butyrate, and also modulated the microbiota of the gut towards more fermentative species like Bifidobacterium, Roseburia, Bloschia, or Lactobacillus. We can see very much the same in a human study where the humans were fed similar diets, so a Western style diet with high in easily digestible carbohydrates, whereas a diet that was rich in fiber has led to the increase of butyric acid in the feces, and also an increase of Bifidobacterium in the hindgut of the pig. So we have in humans as in pigs very similar responses to how the hindgut responds to similar diets. Another study that was really interesting done here at Aarhus University was to look at the plasma metabolome of pigs and human diets, which were both fed with with the same type of bread. So it's the same study as before, where white bread was compared to rye bread and some breads that were rich in some specific types of fibers. So 30 minutes postprandial blood was collected from both the pigs and the humans and subjected to a non-targeted metabolomics approach. What you can see is that they have the same bread patterns, in quotes, the same response to ingesting these breads, the pigs and in humans, where you can see different differences between the white bread compared to the rye bread and other type of high fiber breads. And in on the left side here, you have the pig response. On the right-hand side here, you have the human response. In the humans, the white bread response is actually a bit more accentuated, and it differentiates better from the other types of bread that were used in this study. Another difference that you can see in this plot here is that there's a much higher variation amongst the human subjects compared to the pigs. So that's kind of a difference, but also something very interesting when we want to compare pigs and humans, because of course in a pig study you control much better the responses and what's happening to the pig. You know exactly what is being fed to the animals. You know what is left out of after the study. Whereas with a human intervention study, there could be many factors that affect the metabolism and also digestion. So there could be interferences from other foods that the humans have, or things like environmental issues like smoking or environmental exposure. So there was significant differences between the species, where pigs, as you can see, so there is a big difference between pigs and humans metabolome, where you have higher concentrations of betaine, chlorine, some lysophosphocholine, uric acid, bile acids, creatinine, tryptophan, some amino acids that are much higher in the pigs compared to the humans. And on the other hand, humans have higher levels of carnitine, phospholipids, and other lysophosphocholine. So 19 metabolites out of 26 were detected to be non-significant diet species interaction, but the response was very similar to the type of bread that was used. So in my studies, I have focused a lot on pig obesity models and the use of fat and carbohydrates. And in the study, I want to briefly show you that the ad libitum feeding of a high carbohydrate, high fat diet resulted in a very fast and accelerated development of obesity in this Yucatan model mini pig. We also observed that through CT measurements, an increased deposition of subcutaneous fat, which is representative basically of how the pig deposits this extra energy in an ad libitum context. Of course, there's all of these fat deposition also comes with some side effects on the systemic responses, and we saw an increased cholesterol level, also HDL level and LDL level, as well as increased glucose that was eliminated in the urine of ad libitum mini pigs. And this compares and is a very similar response to what we see in humans, where there were obese humans being checked for all of these parameters. As the obesity progresses, you get a higher increase in a lot of these triglycerides, cholesterol, glucose, and many other parameters that we can also assess in the mini pig model. Another project that I was involved in, and also related to the development of a miniature pig model, we wanted to check in this pig if we can develop some specific signs of obesity and metabolic syndrome, and these would be accelerated by the ad libitum feeding of fructose. And that comes from kind of another story, is that many studies so far have found that the increased intake of fructose, at least in humans or in teenagers, have led to the increase in obesity development, mainly because it's a very easily digestible carbohydrate, and it's a carbohydrate that's being used a lot, like for example in sodas or in fast food type of meals. So we had these Göttingen mini pigs, which arrived at our facility at 8 weeks of age. And after an adaptation of 2-week period and the diet transition, we kept them on an ad libitum feeding strategy for 20 weeks. And at 4 weeks, 12 weeks, and 20 weeks of the dietary intervention, these animals were anesthetized, and we collected blood samples as well as feces, urine, muscle, liver, and also fat biopsies. And every 2 weeks, we recorded feed intake and also anthropometric measurements, so measurements of the waist, of the length of the animal, of the chest measurement, also body weight, of course. So what we saw was, of course, the animals grew and developed obesity severely, but looking at insulin resistance, which is an important factor related to the metabolic syndrome and diabetes development, was that we have all of these known biomarkers related to insulin resistance that were increased during the intervention period with a slow constant increase in glucose, fructosamine, insulin as well, and also C-peptide and glucagon, all of these markers for insulin resistance. We also saw that with the use of fructose in the diet, so this easily digestible carbohydrate, we saw that some biomarkers were increased when compared to the other diet, such as glucagon, for example, or also insulin when measured in the blood. When it comes to dyslipidemia, so like cholesterol, HDL cholesterol, we saw no changes in general throughout the trial, except for this HDL cholesterol, which decreased with time. In this experiment, we also had opportunities to do some measurements that are not done usually in human experiments. So particularly liver and fat biopsies. We collected these samples for gene expression, and this is something that in a human trial you will not be able to do because, of course, you can't biopsy a liver if there's nothing kind of wrong with it. So several genes in different tissues were analyzed, and one interesting response was related to adiponectin. And adiponectin is a known hormone strongly correlated with obesity, and also a decrease in adiponectin is related to insulin resistance and hyperinsulinemia. So we observed a decrease in the PPR gamma expression, which over time is a known key regulator in the adiponectin expression. And we believe that this decrease in the expression of adiponectin in this subcutaneous adipose tissue and also the decrease of the receptor itself in the liver and adipose tissue, they're all connected, probably leading to a sort of a adiponectin resistance and insulin resistance. Obesity, of course, and the development of visceral adiposity is an important role has an important part in the development of inflammation of the system. So we looked at some systemic markers and inflammation in the plasma of the animals, which they were not significantly different when we looked at the systemic inflammation. But when we looked at the at some tissue gene expression markers, for example, IL-6 and chemokine ligand 5, both of these increased with time in the liver. And also CCL5 and also CRP in the liver, they responded differently to the diets, indicating some signs of liver tissue inflammation with the intake of fructose. So earlier I mentioned something about metabolomics, and also half of this presentation today is focused on the use of metabolomics techniques for animal research. The concept of metabolomics is not new. It's been there for many, many years, and because diseases or any external factor that affects metabolism changes the metabolism, and we can see these changes either in urine or in plasma, feces, saliva, any kind of biofluid. For example, like an easy example would be the diabetics excrete sugar in their urine, and this you can actually smell that the urine has a sweet smell, what's called a maple syrup urine disease. There's also an example from almost 5 centuries ago from the urine wheel of Pinder, which is a physician from I think Germany, and he made this chart where different colors of urine and different smells of urine and different things that he could see in the urine were correlated to some kind of affection or ailment. Metabolomics is a part of the omics methods together with proteomics and transcriptomics and genomics. But as a definition, metabolomics is the study of processes involving small metabolites or small molecule substrates, intermediates, and products of cellular metabolism. When we look at genomics or transcriptomics, they are sequence-based analysis. So you look at the sequencing the base pairs, whereas proteomics and metabolomics, they're mass spectrometry based. So you measure masses of these molecules in your sample. And the collection of small molecules and their interaction in a biological system is known as the metabolome. There's two types of metabolomics and different applications. For example, non-targeted metabolomics, which is the analysis of many metabolites at the same time without bias. This is an explorative technique. It also is a hypothesis generating technique. On the other hand, you have targeted metabolomics, which is looking at a predefined set of metabolites measured in samples, and these are usually also quantified. So this is a hypothesis-driven sort of analysis. There's many places where metabolomics is being applied and used. Amongst them, for example, toxicological or pharmacological studies, because if you use some kind of drug or something like this, you want to look at how the metabolome is affected or where exactly is this drug that you use targeting different pathways. You can use it as a diagnostic tool in type 2 diabetes and other diseases that cause changes in metabolic pathways. You can use it to screen for different cancer types, and of course also for nutrition, so as biomarkers for nutrient intake, or also physiological markers if anything changes based on the nutrition. An example here with obese type 2 diabetes patients, you can distinguish them from control patients. So the type 2 diabetes makes a change in the plasma of the subjects. And this is a non-targeted approach, as in you look at as many metabolites as possible. But for example, here there's metabolites of coffee intake, and you specifically quantify. So you have the coffee intake, but you can look at caffeine, trigonelline, or some other metabolites that are specific to coffee intake. So how do we measure this, or what do we measure? We can use different techniques such as liquid chromatography, gas chromatography, NMR. But in LCMS, so liquid chromatography or gas chromatography, we measure intensities of the peaks separated chromatographically, which we used the intensity of these peaks for statistics later on. But also at the same time, we measure the mass of each compound. So this we can use for compound identification later on. So in this non-targeted approach, we don't know what compounds are detected until later on. Everything is basically a puzzle game. We have to make sense of what's happening. So I want to bring some examples from some of the studies that I previously mentioned. One of them was called nutriomics, and we work with Yucatan mini pigs. So 17 of these Yucatan mini pigs were kept on a base diet until 5 months of age, after they were ad libitum fed or restrictive for 20 weeks. And at the end of the experiment, the animals were euthanized, and serum, urine, liver tissue were collected for biomarkers of metabolic syndrome and non-targeted metabolomics. So what we saw was that in all of the samples that we collected in serum, in the liver and urine, there were big differences between the metabolome profile of ad libitum fed animals versus restricted fed animals. So if we go to look into what exactly was changed, we saw that some metabolic pathways were affected by this intervention. For example, pantotenic acid, and the CoA biosynthesis was changed in serum, but also in the urine, with much higher levels of pantotenic acid in the ad libitum feeding. Several amino acid pathways were also affected in plasma and urine, such as tryptophan or lysine. And in liver, there was an increased presence of glutamic acid and glutathione in the ad libitum fed animals. Another example is to the MERIT study, which I showed earlier in the development of obesity in the Göttingen mini pigs. We wanted to see if any metabolic features show up as affected by the time of exposure, so as the obesity progresses. We had this multi-block PLS model where we statistically combine different data sets, so the plasma metabolome with the feces metabolome and urine metabolome, to try to get a better understanding of all of these different compartments on how they change with the development of obesity. So we could see that there was a good separation and transition from week 4 to week 20, and good separation between 4 and 20. However, the PLS model was not able to differentiate well the week 12 from the rest of the group. So this tells us that there is a period in the transition phase between these 2 points. But what is actually happening is that there's many metabolites that change during this period. So like amino acids that increase during obesity development, amino acid intermediates, some hippuric acid or urine conjugates in urine, of course, and in feces, there's a lot of steramides, fatty acids, oxylipins that increase with time during the development of obesity. Another way to look at what was happening during this time was to we tried to see if we can predict body weight using this metabolomics data. So we tested all of the blocks, like the plasma, the urine, feces, and plasma metabolites were the best to generate a model that was statistically significant and had a good correlation with the body weight. And we saw metabolites that correlated really well with body weight, such as leucine or tryptophan. We also wanted to look in more depth at the gut metabolome and also microbiota. So in order to get more information, we combined different data sets from different analytical techniques. So we had feces metabolomics untargeted. We had feces short chain fatty acid. That was a gas chromatography targeted analysis. We had microbiota by 16S sequencing. And we combined all of these into one model to see if we can get any information out of it and see if the diets separate. And in this case, it did. And we could see that there were specific metabolites and specific bacteria, as well as specific short chain fatty acid that all correlated to one another and went in the same direction. So with the use of this high amylose maize starch, there was increase in some ruminococcus. There was higher bacterial fermentation producing succinic acid, pantotenic acid, some isovaleric, isocaproic acid, but as well as the main short chain fatty acids such as acetate or acetate propionate and butyrate. On the other hand, fructose intake was associated with a higher increase of specific bacterial feces that specifically consume fructose or simple carbohydrates. And also we saw some metabolites that are more specific in this case, probably caused by these bacteria. So it's been a bit longer than I thought, but some take-home messages. Pig models are great substitute for human research, but the use of it depends a lot on the question asked, and are we going to be able to keep developing these new models? Are we going to be able to use them in the future? And I'm asking this question because we're probably going to face a lot more restrictions in the future with the use of animals in research. But they can provide high value for questions where humans cannot be used directly, or where procedures are not allowed in human studies, such as these, for example, liver biopsies that we did. And metabolomics is an excellent tool for exploration and looking for unknown changes given by a diet or environmental factor. It boils down a lot to choosing the right method depending on the study and the question asked. So are you going to do a targeted study? Are you going to do a non-targeted study? The targeted methods and quantitative methods, they're really nice because they give you a deeper understanding on metabolite fluxes and on metabolic pathway changes. But of course, the untargeted, you can see things that you might not expect to be there. And then just as a last point, choose to combine with other omics methods, or combine data sets in a multi-block analysis method, because it gives more biological meaning, and you can cover a lot more information. So that was all, and thank you all for your attention. I'm open for questions if you have any.
So thanks a lot, Mihai, for your very complete and informative presentation. And are there any questions from the participants to Mihai? Maybe I can start with a question. Oh, sorry, there is one from Ines.
Yes, I have a little question, because you talk about using pigs for human model, but you also talk about different breeds. You talk about Yucatan, Göttingen hen, I think. And I imagine that there's also like industrial breeds. And is there a difference between all of these? Because genetically, they are completely different and in size and everything.
Yeah, so again, the choice of the pig, I think, boils down a lot to what you are looking for, or what the experiment is, or the research question is, because I would say in my specific case, in my examples, we were looking for a model that's specific for obesity development and metabolic syndrome development. And for this kind of work, miniature pigs are best, because they grow when you have such a diet, when you feed them ad libitum, they're gonna grow crazy, and you can't do this with a commercial breed. You can, but it's almost impossible to handle such animals after one point. So the choice of using miniature breed is to keep the same response of the metabolism, in terms of developing obesity, in terms of developing some cardiovascular issues, or changes in the blood work, but the size will be manageable in the end.
Okay. Yeah. Thank you.
Yes, I have another question, because you are speaking about the advantage of using the mini pigs, but do you see also any challenges or limitation for using them?
Yeah, there's quite a few. It depends also on the breed that you work with, or you select to work with. From the experience I've had, Yucatan mini pigs are very temperamental, we could say. They're not easy to handle, so they have a bit more of a wild being. So it is actually difficult to work with in practice in terms of like, I don't know, placing a catheter and trying to sedate them or handle them if you want to work with them in some procedures that require them to be awake. For example, in my experiments, we collected some saliva samples, and it's trying to handle the animal in a way that it doesn't stress them, but at the same time you can still collect your sample. And with Yucatan is so difficult because they stress very easily, and actually there's been cases where you can stress them like so much that they die. Like they're very prone to having heart issues, so they can easily pass away if you really stress them. And with miniature pigs, that becomes easier. Their temperament, so to say, is much more calm. It's much more allowing, and you can train them like really easily. Like they're very much like pet dogs that you can train them from very little, that they won't, that they'll be very cooperative. So that's some of the challenges in terms of handling the animals. There's also challenges in terms of sample collection, for example, because when they're too small, like for example, this little guy that's in the picture, you can't do much sample collection when they're at this stage. So there's so little, and it's very dangerous to, depending on your analysis, of course, but if you have to take so many samples, then it's very challenging. That's why in the mini pig, Göttingen mini pig experiment, we have sampling starting at only 4 weeks after the diet. So not in the very beginning, because they were just too small. There's, things like this.
Yeah. Thank you. Yeah.
You're welcome.
Hi, Mihai.
Yes.
Thanks for the presentation. Well, I have 2 questions, but I had one, but from your previous comments, I found it. I worked with Yucatan mini pigs during some of my experiments, and I found it the inverse.
Okay.
For me, they were very kind. They were very easy to handle, much more easier to handle than the let's say regular pigs.
Okay.
And I was wondering, is it because of maybe it's because of the age, because you were working with obese pigs, and I imagine since you use it for human experiments, they were much older. My pigs were like less than 1 year, 11 months, 10 to 11 months.
No, ours was also around like 5 to 6 months of age, so young,
Oh, okay, and they were already obese.
With the right amount of diet and a few weeks on ad libitum feeding, they get very obese very fast. Yeah, but I don't think that will drive any kind of difference in the behavioral in the behavior. I actually, probably the more obese they get, the calmer they get in the way, because they're not going to be as active. No, but it's true. It's a bit unfortunate, but they're not going to be as active or as intense in a way. But I don't know, it's just maybe our experience here with the Yucatan in particular, that they've been a bit more challenging, especially for blood samplings and things like this. Maybe they weren't trained as well. I'm not sure. But especially like females, they were challenging.
Okay. Did you sample blood by jugular vein or by catheter? Okay. Maybe that's why. Because my pigs were catheterized, and they were, to be honest with you, they see me every day. And some pigs, when I come in their pen, they even lay down to have me scratch their bellies. So that's why I said, for mini pigs, I always thought they were always like that.
That's the same experience I had with the Göttingen mini pig. So it's when I say that there is differences between the breeds themselves and how they behave and how they react. So mini pigs are exactly the same as you described. Göttingen, sorry. But the Yucatan, on the other hand, a bit more tricky. Yeah.
Okay. Yeah. Well, so onto my main question then. You mentioned something at the end in your take-home message. should we continue these types of experiments with pigs, especially with maybe incoming regulations or maybe possible changes in the future? And I just wanted you to comment a little bit on that, because if we're not able to do this in pigs, do you think it should be done on other species? Or if it really is not done in the European community, do you think it would just transfer to another country or someplace else?
That's very good questions, all of them. It's tricky to say, but I mean, you know as well that we have these pressures that probably we're going to have to limit a lot more the use of animal in general. So now we're having a specific case, more special case, where we talk about them being used as biomodels for human research. And I think maybe in a way that could be seen as like protected, you know, because if you define your scope very well, that your animal model has benefits and has something that's very important to the human, it, as it is so far, it bypasses a bit easier. So, you know, you talk about animals to be used in research. We think about like nutrition research and everything that we do in general, you know, with production animals, and that's where the pressure comes from. In this case, it might be a bit saved by the fact that we're trying to use them for humans, human purposes, or learning more. And yeah, I think it's a very tricky area, but probably what's going to happen is maybe try to go more into like models of like organs or cell lines. So I see that happening a bit more rather than using the whole animal. But yeah, I think it's going to be a challenge. Yeah.
Okay. Just one last thing. Last week I was talking with someone from Norway, and actually his opinion actually is the inverse. He said that he thinks that using animals for human models would be actually banned, because they said that humans can give their consent, whereas animals can't. Like, I mean, if you go to a clinic and they say, we're going to do this study on you, do you agree? You sign. That takes away all of the let's say pressure from the experiments.
Yeah.
But for animals, they don't have a choice. But I don't know. I find it, yeah.
I see
Anyway,
that point. I see that point, but I could also say like there's going to be so it's all about the specific case, right? Like if you're going to look into liver disease and you want a model for liver disease, and you take the pig, for example, then you can do a lot of things that you're not going to be able to do in humans. And then the issue of consent doesn't even exist, because there's not going to be a doctor ever that's going to ask a patient, are you willing to go on the table and have your liver biopsied for my little experiment? That's not going to happen, because that goes kind of over their medical like thinking that you can't kind of do something in a healthy patient. Yeah. So it's yeah, I can see that viewpoint, but it's not I don't think it's going to be completely banned.
Yeah, I agree with you. Thank you.
Are there any other questions?
For me, I don't have anything else to add, but just the next webinar should be after the summer.
Yes, it will be in September. It will be Jacinta Bus who will speak about feeding patterns, but we will announce the webinar.
Yeah, we will announce it when, Okay. Okay. Thanks, everyone.
Thank you.
Thank you.